The TIPS program in next generation vaccines is really about two revolutions that happen in parallel in our field. One is in structural biology. We now can actually observe how viruses attach to the human host cells, and we can also observe how antibodies that the human immune system produces interfere with this process. The second important piece of information is that for the first time we are able to do high-throughput sequencing on the human antibody repertoire. So we have a very clear understanding of the millions of antibodies that the immune system of a healthy person produces, and also how this antibody repertoire changes if a person gets infected or vaccinated.
Conventionally, vaccines have been made empirically by weakening a virus or bacterium and injecting it into animals to see if it works and induces an immune response. And vaccines have been the most important biomedical advance in history. We've eradicated several diseases with that strategy, but now we find vaccine development is going slowly because the germs we're trying to target are much more wily and difficult. So we're looking for a new approach to vaccine development that will accelerate our discovery and our ability to understand how vaccines work and how to develop them safely. Vanderbilt's an exceptionally good place to do vaccine research because we're a world university. We're connected to so many international sites. We have both a strong university and a medical center, and our medical center has particular strength in global health and international activity. So we're able to go all over the world with colleagues and collaborators and find individuals who have experienced infections and to work with them to obtain samples that will help us in our research. We obtain the blood wherever the person lives, anywhere around the world, and we're able to process that blood sample and obtain the immune cells from the blood, separate those out and freeze them, and we can ship them back to our laboratory here in Vanderbilt in Nashville.
I'm Jessica Finn. I'm a graduate student in the Crow and Myler laboratories. Okay, if you think about all of the diseases that you need your immune system to protect you against, you kind of pretty quickly realize that's a big problem. The immune system has to protect you against many different potential infectious diseases. You don't just have one or two antibodies. You have antibodies against every single infectious disease that you've seen or infectious diseases that you may be infected with in the future. So we develop these computational screening methods that allow us to look through that database of sequences and find the ones that could be specific for diseases we're interested in studying, like influenza or maybe HIV or Ebola. And then we can take those antibodies, express them in the laboratory, and test whether or not they would be effective at protecting us against those diseases.
To realize this vision, we need a lot of really recent novel technologies that we actually get from basic sciences. So this is a collaboration between the Vanderbilt Vaccine Center, which worries about patient care, and the Center for Structural Biology that actually focuses on developing these technologies to understand the atomic detail of this process of infection that develops the computer algorithms to design these antibodies.
So in the TIPS project, we are getting proteins from the Crow lab where they have made antibodies that are neutralizing towards a certain antigen. So they give us both the antibody and the antigen, and we make a complex of that, and then we use structural approaches to generate a model for how they interact. So Vanderbilt has a really great Center for Structural Biology, And the whole goal of this center is to make structural biology accessible to everyone at Vanderbilt. So we have techniques for doing X-ray crystallography. We also have a large number of NMR machines, but those are for smaller complexes. And then we have cryo-EM, which is what I'm an expert in. And my lab used cryo-EM to study the shape and the way molecular machines look that we purify from the cells. And for the TIPS projects, what we really want to understand is how the antigen and the antibody are interacting. Because if we can understand how a neutralizing antibody is actually interacting with the antigen, then we may be able to design one that's even better.
My name is Alex Sevi. I'm a graduate student in the Crow and Myler labs. Generally what I work on is using computational models to integrate information about antibodies and figure out a little bit more about how they work. So we take information like from electron microscopy and X-ray crystallography, and we combine those using computer models to really drill down and figure out exactly how are these antibodies working. We can use these models to specifically point out where are they binding to their targets, how likely are they to be resistant to future viruses that may come up in future years, and how good of targets would they be for some type of therapeutic or for some type of drug, or potentially for developing a vaccine for that particular virus.
So it's really a project that goes all the way from basic science, where scientists develop these methods and learn how to apply those to these questions, into the medical school, into the clinics, where we can make those antibodies and test them and ultimately develop vaccines that could be used in humans. This program involves 10 faculty from the Vanderbilt Vaccine Center and the Center of Structural Biology that really need to work together, bring together a large number of different technologies from both fields to be successful. And so it's a highly interdisciplinary program that really only functions because these faculty are so close in space and can collaborate easily. We not just focus on making those antibodies and designing the vaccines, we actually have a program that educates graduate students and postdoctoral fellows. I mentioned this is a very interdisciplinary project, so experts from different fields, structural biology and immunology, have to collaborate. Each student, each trainee in this program has a mentor from the Center of Structural Biology and a second mentor from the Vanderbilt Vaccine Center. And each trainee is trained in technologies from both sides. We believe that in the future, to continue this research and populate this emerging field with researchers, we really need to change how we educate people. We try to not just pioneer this field, we also try to simultaneously train a generation of leaders for this emerging field. And we expect that these trainees will move on to industry and or academia to really take this interdisciplinary approach that we are developing here out into the world and make it work on many of these very important research projects.